Welcome to The Explainer. Today, we're doing something really cool. We are shrinking way down to step inside the ultimate chef's kitchen. I'm talking about the microscopic world of biology. It is absolutely fascinating how every single living organism out there is built from just a handful of basic ingredients. And the way these ingredients interact, well, that creates the absolute mind-blowing complexity of life. It's this brilliant, never-ending system of building things up and breaking them down.
So, think about the last meal you ate. Got it? Okay, how long do you think it would take your body to completely digest that food if you didn't have the help of some very special biological machines operating inside of you right this very second? Any guesses?
The contrast is honestly mind-blowing. Without these biological machines, which we call enzymes, it would literally take your body around 2 to 3 weeks to digest just one single meal. 2 to 3 weeks. But with enzymes acting as these hyper-fast prep chefs, it takes around 4 hours. So without them, metabolic reactions just simply couldn't happen fast enough to keep us alive. So here is our roadmap for this explainer. 1. Building blocks of life. 2. The big 3 molecules. 3. Playing food detective. 4. Meet the enzymes. And finally, 5. Temperature and denaturation. Let's dive in.
Sections 1 and 2. Building blocks and the big 3 molecules.
You know, it's pretty amazing how most of the molecules found in living organisms fall into just 3 main categories: carbohydrates, proteins, and lipids. And because they all contain carbon, we refer to them as organic molecules. But they aren't just carbon. We've got oxygen, hydrogen, and occasionally things like nitrogen and sulfur joining the mix to build the foundation of basically everything.
A great way to think about carbohydrates is to picture Lego bricks. You start with small, simple sugars, called monosaccharides, like a single unit of glucose. But when you start snapping lots of these simple glucose molecules together, you build these massive, complex, large molecules called polysaccharides. And because these really large chains are insoluble, they become incredibly useful to the organism as storage molecules.
Now, what's really interesting here is how this clears up a pretty common misconception about these storage chains. See, even though both plants and animals use chains of glucose for storage, they actually do it differently. Plants store their glucose as starch molecules. Animals, on the other hand, store glucose as glycogen molecules, which you'll very often find tucked away in animal muscle and liver cells. So it's the exact same basic building block, just a completely different final structure depending on the organism. Moving on to lipids, we see they're built a little differently. Instead of those long, repeating chains like the carbs, they form from smaller basic units. Specifically, you've got a single glycerol base attached to fatty acid tails. A really common one found in the human body is a triglyceride, which is literally named for the 3 fatty acid tails linked right to that single glycerol backbone. And finally, we have proteins. Like the others we've talked about, proteins are large molecules formed from smaller units, but here those units are called amino acids. And if you arrange these in different orders, you get hundreds of thousands of different proteins. The absolute key takeaway here is that a protein-specific sequence creates a very unique shape, and that shape dictates its exact function, whether that's the keratin in your hair, the hemoglobin in your blood, or, as we'll see very shortly, our enzymes.
Section 3. Playing food detective.
Okay, so before running any actual chemical tests, you have to prep your sample safely. Because, well, you can't just go pouring chemicals onto a solid chunk of food, right? First, you break it up using a pestle and mortar. Then you transfer it to a test tube, and you add some distilled water. After mixing it all really thoroughly with a glass rod, you filter out the solid chunks. What you're left with is this nice liquid solution containing all the molecules ready for testing. And hey, safety first for sure. Always wear your eye protection and absolutely never taste the substances in the lab.
Let's take a look at one of the more involved lab procedures, the Benedict's test for glucose. So you add a few drops of Benedict's solution, which naturally starts out light blue, right into your sample. But here's the catch. This test requires heat to work. You actually have to place the test tube into a beaker of very hot water for about 5 minutes. And as a super important safety precaution, if you used a Bunsen burner to heat that water up, you must turn it off to remove the open flame hazard before you proceed. If glucose is present, you are going to see a really dramatic color change.
So this right here is essentially your ultimate detective cheat sheet. It summarizes all the magical color changes you'll see. For glucose, that heated Benedict solution turns from light blue to a bright orange or brick red. To test for starch, you use iodine solution. If it's a positive result, it shifts from yellow-brown to a really deep blue-black. For proteins, the Burat solution changes from blue to this vibrant violet or purple. And for lipids, shaking the sample with ethanol and then adding cold water will turn your clear liquids into a highly visible cloudy emulsion. It's literally chemistry revealing the invisible building blocks of our food.
Section 4. Meet the enzymes.
All right, let's zoom back in on the microscopic action. Enzymes are highly specific. The substance that an enzyme acts upon is called its substrate. Now, in a solution, these enzymes and substrates are just randomly moving about. And when they randomly collide, they snap together to form an enzyme-substrate complex. The enzyme has this really special area called the active site, which happens to be the exact complementary shape to the substrate. They fit together perfectly, literally, like a lock and a key. Boom! The reaction happens, breaking the substrate into products, which then float away. But the absolute best part? The enzyme itself is completely unchanged and is instantly ready to grab the next substrate.
Now, because enzymes are proteins, their specific sequence of amino acids dictates a highly specific shape. It quickly becomes clear that the shape of the active site is its single most critical feature. If it doesn't perfectly match the substrate, that whole lock and key mechanism completely fails. And without that fit, those life-sustaining reactions we talked about simply won't happen.
Section 5. Temperature and denaturation.
Let's start with the ideal scenario here. Enzymes work fastest at what scientists call their optimum temperature. In the human body, that optimum temperature is exactly 37 degrees Celsius. This is the absolute sweet spot. At this temperature, molecules have enough kinetic energy to move around rapidly, ensuring we get plenty of those vital collisions between our enzymes and our substrates.
But heating an enzyme far beyond this optimum temperature, well, that introduces a massive problem. High heat physically breaks the delicate bonds that hold the enzyme's complex protein structure together. Once those bonds break, the enzyme literally loses its shape. And if the active site loses its specific shape, the substrate can no longer fit inside. The lock is busted, and the biological machine is completely broken.
Scientists call this destructive process denaturation. And the really important takeaway here is the finality of it all. Denaturation is irreversible. Once an enzyme is denatured by high heat, you can't uncook it. It cannot regain its proper shape. Its biological activity stops entirely. It's a permanent breakdown in the system.
Now you might be wondering, what about the cold? Well, it's a vital distinction to make here. Low temperatures absolutely do not denature enzymes. Cold doesn't break the chemical bonds or destroy the active site's shape. It simply reduces the kinetic energy in the system. The enzymes and substrates just move much more slowly, which means they collide way less frequently, ultimately slowing down the rate of the reaction. But the key here is that the enzymes are still perfectly intact. And, that leaves us with something really fascinating to ponder. We've just seen how easily enzymes can be permanently destroyed by heat, and yet, they are the very things keeping us alive. So if our internal biological machines are this incredibly vulnerable, how on earth does your body manage to perfectly regulate its internal temperature at exactly 37 degrees Celsius every single second of your life? Well, it's a brilliant biological balancing act called homeostasis, and it's the perfect topic for us to dive into next time. Keep asking questions, and I'll see you in the next explainer.